Hello and welcome to my presentation about my web tool of choice, which is tRNAscan 2.0. The publication that I used as a source of information to better understand the web tool and how it works is displayed in this slide. This publication was made by some of the people who worked on and created the web tool, so it's a pretty reliable source. So the goal of the tool is to identify tRNA genes encoded in genomes. It can also detect tRNA-like structures or sequences that have relatively high similarities to tRNA genes. As a means of introduction, I will shortly explain what tRNA is and what the context of it is in the translation of proteins. So in the left below picture you can see how a ribosome uses tRNAs that have a complementary anticodon to the mRNA that needs to be translated into protein. Attached to the tRNA is a amino acid that binds to this tRNA isotype. In the right image you can see an example of a secondary structure of tRNA with clearly visible the anticodon in close proximity to the mRNA sequence. The first thing you'll see on the input page of the web tool are the search options for the sequence sources. With these options you can choose a domain-specific model on tRNAs exclusively from, for example, eukaryotic or bacterial organisms. Doing this, you can accelerate the search speed without affecting the sensitivity of the search. You could also choose the mixed option, which is basically all the tRNA models put together, but this however will slow down the search. You could also choose the mitochondrial options if your sequences come from a mitochondrial genome. The search modes that are available are default, legacy and infernal without HMM. The default one uses infernal 1.1, which is a software that I will explain in the next slide. Legacy is basically the older version of the tRNAscan software. This legacy option gives the availability to do backward comparisons with the newer version, even though this shouldn't really be necessary as the results should be the same. Another difference, however, between the legacy and the default search mode is that the default one can detect chimeric tRNA, which are basically point mutations in the anticodon of the tRNA. Legacy can't find these, so that's another important difference. The infernal without HMM option is meant for short, low-scoring tRNA-like sequences that have lower similarities, because this will increase sensitivity, but it makes the search very slow. This search doesn't use HMM profiles, and because of this it's a lot more sensitive, like I said earlier, but at the same time also a lot slower. So the default search mode uses the infernal 1.1 software. What this does is it builds probabilistic models called covariance models of the conserved sequence and secondary structure of an RNA family. This might sound familiar to some people because it's actually a lot like the hidden Markov models. It's actually the exact same, but with the added complexity necessary for modeling RNA secondary structures. Using this software compared to the legacy search mode, it makes the search speed go 100 to 10,000 times faster. This is the part of the input page where you can upload your FASTA file or multi-FASTA files. You could also just copy paste the sequences in the given space. So now I will try to explain what the search software does while it's working. But before I can do that, I have to explain what tRNA isotypes are. So a tRNA isotype is basically a subgroup per complementary amino acid. The complementary amino acids come from the type of anticodon that the tRNA has. So in total there are about 20 isotype tRNAs, because there are 20 amino acids in total. While the search is going, there is a initial first pass scan and then a second pass scan. During the first pass scan, Infernal is used with a permissive score threshold of 10 bits in combination with a search model that traces all the tRNA isotypes in order to obtain high sensitivity. A second pass scan uses the candidates from the first pass scan, but with a higher score threshold to increase selectivity, and covariance models are used for each of the 20 tRNA isotypes instead of all of the isotypes together, like in the first pass scan. When the search has ended, the results can be analyzed. Here I will discuss the most important results. Similar or identical tRNAs can be found in the genomic database. As a lot of tRNAs have already been identified, it's certainly interesting to check if the found tRNAs are unique, or if there are identical results. This can be considered an extra proof that correct samples were sequenced. The predicted tRNA secondary structure can also be viewed as an image in the results. It's also important to compare the Infernal score from the first scan with the isotype score from the second scan. Why this is important will become clear during the demonstration of the webtool. The tRNA isotype is predicted in two ways. The first way uses the predicted secondary structure of the tRNA to locate the correct anticodon. Based on this anticodon and its compatible amino acid, the corresponding isotype is selected. The second way is the result of the second pass scan. During this scan, all 20 isotype-specific covariance models are used, and the highest scoring one is considered as the correct isotype of the tRNA. If these two isotype predictions don't line up, it's reported in the results for further user inspection. In most cases, this is caused by chimeric tRNAs which have a point mutation in their anticodon. Now for the demonstration, I will describe the research question in order to have a practical setting to use the webtool. Adding more annotations to mammalian genomes for non-coding RNAs for animals such as the house mouse, which is a relevant research model, is important to further understand the organism. Find the non-coding tRNA genes in the mus musculus IRG locus from chromosome 11. So now I will use this accession number to find the locus. Then you just save the FASTA file, create file, so it's called the ERG locus. Then we can go to the tRNAscan site. Here you can, like I explained it, choose your sequence source, we will choose eukaryotic, since we use the house mouse. Then we use the default setting, which is the most logical one. We don't need the older version, and the infernal without HMM one was for shorter sequences that have very low similarities. So maybe if we don't have any hits with the default one, we could retry it with the infernal without HMM filter. But now we will use the default one. So we upload our file, which is here, and then we click on output. So these are the results. 9 hits are visible. If we look at the most right column, we can immediately see if we have good hits, really bad hits, or hits that indicate something else. We can see nodes that say none and then nodes that say pseudo. The pseudo nodes basically indicate that these are pseudo tRNA sequences. This means that these sequences are tRNA-like sequences that have similarities to the real tRNAs, but they themselves are not tRNAs, and they also don't have a tRNA function within the organism, but they do have high similarities. So we can see that the ones with none are in fact good tRNA results. These have a very high probability of actually being tRNAs. And if you look at the second hit, which is this one, then, and if you look at the infernal score and the isotype score, then you can see logical scores. The infernal score is relatively high, it's 78, and while the isotype score is even higher, it's 93. And this is logical because the infernal score is based off the first scan, where all the tRNA isotopes are used together in one model. So this is less specific, and as it is less specific, the score is lower. However, the isotype score is the score of the best scoring isotype model. So 20 isotype models were tested, and the best model with the best score is the one with this amino acid and the complementary score of 93.4. So if we now take a bad example, for example the first hit, which is a pseudo tRNA that was found, we can see that the infernal score is actually higher than the more specific isotype score. This basically means that a more general scan gives a better result than a more specific scan. So this further indicates that we are talking about a pseudo tRNA, because if it would be a real RNA, it would identify with a higher score with at least one isotype, but it doesn't, and the reason why is because it's not a real tRNA. After that, we can have a look at the isotype. So there were two ways to identify the isotype. The first way was by usage of the anticodon. The anticodon was located by using the secondary structure, which can be viewed here. So here in this example, TGT is supposed to be the anticodon, and if you look at the structure, you can see UGU, which is basically the DNA counterpart of the anticodon. So this here is the amino acid that is complementary with this anticodon, and based on this amino acid, the isotype is created. Now there was a second way to determine the isotype, and the second way was via the usage of the second scan of the search model. And the isotype score that you can see again is the highest isotype score, and based on this highest score, which you can find here, it's the same 93%, and then the second highest is this 73%, and then the third highest is 72%, but only the highest is selected, and based on this highest score they assume that this would be the amino acid that would bind to this specific tRNA, and based on that the isotype is selected. So in both cases we can see that the amino acid here and here is the same, which indicates that this is a true tRNA and not a chimeric tRNA, because if it would be a chimeric tRNA with a 1-point mutation in the anticodon, then the isotype detection method using the secondary structure could potentially show a different amino acid, while the isotype score detection method wouldn't respond to the 1-point mutation. So other than that, there is still one other important part that has to be discussed, which is the similar tRNA sequences that can be found in the genome tRNA database. Let's take a good example, this one. Then you can see two lists. The first list is a list with perfect matching tRNAs from the database. These have 100% coverage and identity. And then there is a second list with tRNA sequences that have a lower similarity. So these have either a identity that is a bit lower or a coverage that is not 100%. But all of these are still very similar sequences. So then finally we can answer or we can give a conclusion to the research question. There is a very high probability of the tRNA candidates 2, 3, 4 and 9 being real tRNAs, and then the other candidates are basically pseudo tRNAs or tRNA like structures. This was indicated by the infernal score that was higher than the isotype score, and even the infernal score was actually a bad score. So this was my presentation, and thank you for listening.